Hello class, welcome back to genetics. I'm your TA Caroline, and today our lesson is going to be about population genetics. Our model organism for today is going to be one of my favorites. It's the Bowtruckle, these cute little guys. So here you can see I have drawn out the three phenotypes that we will be looking at today, with the dominant allele homozygous representing green, the recessive allele representing white, and then both of the alleles together representing a lime green color. Now, Haggard and I have been monitoring this population of Bowtruckles for a while now. There's about 300 of them, and we have proven that they are living in Hardy-Weinberg equilibrium. So here's our sketch of the population. You can see all the different phenotypes, and these are the number of genotypes that we observed. And with this, I was able to calculate the genotypic frequencies and the observed allele frequencies. So now that I calculated the observed allele frequencies, we can find the genotypic frequencies of the population if they were to be in Hardy-Weinberg equilibrium. And we do that by using the equation that we learned last lecture, p squared plus 2pq plus q squared. Now, each part of the equation represents a genotype. So p squared represents the homozygous dominant, 2pq represents heterozygous, and then the q squared represents homozygous recessive. Now, here are our allele frequencies from earlier. If you just plug those in, our expected frequencies are 0.2827, 0.49796, and 0.2193 for the respective genotypes. Now, with these expected genotypic frequencies, we can conduct a Chisquare test to see if this population that we observed is truly in Hardy-Weinberg equilibrium. Okay, so right here you can see that we have our Chi-square table with all of our values in it. For this test, our null hypothesis is that the population we are looking at is in Hardy-Weinberg equilibrium. So here's our observed and expected. I multiplied our genotypic frequencies by the total population, 300, to get these real numbers. And then after all this math, we get to this chi-square value. And since it's less than our critical value, we failed to reject the hypothesis. The B locus in the Bowtruckle population is in fact in Hardy-Weinberg equilibrium. So when Haggard was looking over the population, he noticed that the dark green Bowtruckles and the white Bowtruckles were dying more often of fungal disease, meaning that the light green Bowtruckles are more resistant. Now, this is an example of stabilizing selection, where the heterozygous is favored over the homozygous genotypes. So with that knowledge, he was able to come up with these fitnesses for each genotype. Now with these fitness values, we can predict the genotypic frequency shifts after a generation. So if you look here, we are able to find the frequency of survivors, even though since this is not out of 1, we need to find an average value to divide it by. So we add all of these decimals together to get this 0.9122, which is our average fitness, and then by dividing each value by this, we are able to find the new frequency. And so as you can see, the homozygous frequencies decrease significantly, while the heterozygous frequency increased very significantly, which corresponds with stabilizing selection. So in the future, the homozygous population is going to die out if this continues, leaving only the heterozygous population. But that also means that both alleles are going to stay in the population.